The overall goal of this procedure is to produce a homogeneous glycoconjugate vaccine by combined non-canonical amino acid incorporation and click chemistry.
Genetic code expansion is a powerful tool to incorporate unnatural amino acids into proteins, to modify their characteristics, to study or create new protein functions, or to have an access to protein conjugates. Codon suppression method have emerged as the most popular method to incorporate unnatural amino acids at defined position. This methodology will be here applied to the production of a protein carrier containing an unnatural amino acid harboring a biorthogonal functional group. This reactive handle can next be used to specifically and efficiently graft a synthetic oligosaccharide hapten to provide a homogeneous glycoconjugate vaccine.
The propargyl lysine PRK is synthesized in 2 steps from commercial bulk lysine. In the first step, the unprotected amino group of the bulk lysine is coupled to propargyl chloroformate. In the second step, the alpha amine group is deprotected. To synthesize the N-alpha bulk propargyl lysine, first dissolve 500 mg of bulk lysine in a mixture of 5 ml of aqueous 1 molar NaOH and 5 ml of THF in a flask and fit the flask with a silicone septum. Cool the flask in an ice bath and wait for the powder to be dissolved. Then, add the propargyl chloroformate dropwise over a 2 to 3 minutes period under stirring. Warm the reaction mixture at room temperature and continue the stirring for 10 hours.